So at Aerie, our major goal is to build a full-fledged ophthalmic pharmaceutical company. Of course, we're starting in the glaucoma market, where we have two products. Both are phase 3 stage products, both are once-a-day medications, and both, if approved, would offer the first new MOAs to come into the glaucoma market in nearly 20 years. Rhopressa is our triple action product. Rhopressa inhibits Rho-kinase, or ROC, as it's listed on the chart. So it opens up the trabecular meshwork to restore outflow through that channel. Rhopressa is also a norepinephrine transporter, or NET inhibitor, so it reduces the production of aqueous humor. And finally, Rhopressa lowers episcleral venous pressure, or EVP, so it provides a unique aspect because Rhopressa lowers IOP the same amount regardless of whether the patient has high pressure or low. That's a differentiating characteristic. Rocklatan is our fixed dose combination which combines Rhopressa with latanoprost, so it adds yet a fourth MOA into the mix. This slide shows the results of our Rhopressa phase 2b trials in which the comparator was latanoprost. You'll see in the upper chart the full cohort of patients with an IOP between 22 and 36. The IOP reduction offered by the latanoprost group is approximately 1 millimeter of mercury better than that of Rhopressa. Importantly, if you look at the chart below that, in our pre-specified subgroup analysis of moderately elevated IOPs, that is 22 to 26, you can see that the IOP lowering was the same between Rhopressa and latanoprost. Now, what caused that? Well, first off, latanoprost did exactly what latanoprost was expected to do. That is, it provided less IOP reduction at lower pressures, where Rhopressa was the same, regardless of whether the pressures were low or high. This is the results of our Rocklatan phase 2b trials. Rocklatan is the gold bars on the very bottom, showing the IOP reduction there, and the two comparators are the components of Rocklatan, that is, both latanoprost and Rhopressa, in the purple and blue bars. The end result of this is the IOP reduction by Rocklatan was statistically superior to latanoprost at each and every of the 9 time points. In fact, the margin of difference was between 1.6 and 3.2 millimeters of mercury, so outstanding performance. So the question now is, what's next out of our R&D area? We're beginning to explore the area of disease modification. The question is, can Rhopressa perhaps modify glaucoma disease? To that, we're looking at 2 questions. The first is, does Rhopressa have any antifibrotic or stop the fibrosis that occurs in the trabecular meshwork? And secondly, could treatment with Rhopressa increase the perfusion that occurs of the trabecular meshwork? Very briefly, this shows a progressive degeneration that occurs in the trabecular meshwork. Once exposed to aging process as well as oxidation, fibrosis occurs in the trabecular meshwork, causing stiffness and shrinkage so that the aqueous humor cannot flow rapidly through the trabecular meshwork. That causes less perfusion of fluids and nutrients into the trabecular meshwork. The concept is if we introduce Rhopressa and stop that antifibrotic activity and increase the perfusion area, perhaps we can change the course of the disease, and we think we can. So we have a couple of studies or very short experiments that I'll show you now that have been recently released. This is done in Dr. Rao's office at Duke, and he was looking to say, could Rhopressa provide any antifibrotic activity in the trabecular meshwork? So these are trabecular meshwork cells. The column on the far left shows the trabecular meshwork cells in the medium. The column in the middle shows what happens when these cells were exposed to a TGF-beta2, which is a pro-kinase cytokine that occurs in the aqueous humor of glaucoma patients in an elevated amount. And when that happened, we got those pro-fibrotic markers to express themselves. The third column on the right shows what happens when 13324, which is the active ingredient in Rhopressa, was then introduced. And you can see they were eliminated or reduced significantly. So that's the first sign that says Rhopressa may actually, in fact, be antifibrotic, which is a very good sign and encouraging us to continue. The second experiment was also done at Duke with Dr. Stamer, as well as up in Boston with Dr. Gong, and they looked at trabecular meshwork tissues, as was shown in the picture there. These are trabecular meshwork from the same patients, and they perfused the trabecular meshwork with either saline or saline in addition to 13324, again, the active ingredient in Rhopressa. Fluorescein beads were put into the liquid so that you could better visualize when perfusion was actually increasing and occurring. And you could see the one on the left that says plus AR13324, there's much better perfusion. In fact, the chart on the right quantifies how much that is, and you could see the area of effective perfusion is twice that in the Rhopressa, or the 1332 forearm as compared to control. So again, this may show that Rhopressa is at least indicative here in these very small studies of stopping fibrosis at the trabecular meshwork level and increasing the area of perfusion. So it's highly encouraging for what might be a disease-modifying product. Thank you for your attention again. Thank you for having us.